Hello and welcome to Unit 6, Lecture 7. In this lecture, we're gonna look at RNA, which is like DNA, but it does a lot of the intermediate work between DNA and proteins. Learning objectives: describe how RNA differs from DNA. Explain how the cell makes RNA. If we compare DNA and RNA together, you can see DNA has that double helix structure. RNA very often is actually a single strand. Okay, so it's almost like half of a DNA structure. It also has ribose, hence the reason it's called RNA or ribonucleic acid instead of deoxyribose. And then RNA is generally single-stranded instead of double-stranded. And last but not least, we get uracil instead of thymine. So in DNA, you have A, T, G, and C. In RNA, you have A, U, G, and C. When it comes to protein synthesis, there are three main forms of RNA that are involved. There's the messenger RNA, there's transfer RNA, and there's ribosomal RNA, so mRNA, tRNA, and rRNA. They all do something a little bit different because DNA, the one that's running it all, can't get out of the house, can't get out of the nucleus. Okay, and so what happens is it needs to use these different RNA molecules to do the rest of the work, if that makes sense. So the messenger RNA is called is mRNA. So that's the messenger. It actually gets made in the nucleus, takes the message from the DNA, and actually carries it out into the cytoplasm. The tRNA, or transfer RNA, is the one that actually carries the amino acid. So it uses the amino acid, or it uses the code to tell it which amino acid that it needs to bring. And they match up kind of like a puzzle piece, but it puts the correct amino acid there. And then last but not least is ribosomal RNA, or rRNA. Ribosomal RNA is it basically makes the tabletop where all of this happens. So the messenger RNA and the transfer RNA actually meet up at the ribosome, or the rRNA, okay, to finish the process, to do step 2, which is actually going to be in the next lecture. That's so you know all the different types of RNA. So messenger RNA is mRNA that's actually used to make the protein. So what happens with mRNA, okay, uses the DNA, right, okay, as a guide, as a template. Basically the same way that DNA goes through replication, DNA is used to actually make messenger RNA. It's very similar, okay? And so what happens is inside the nucleus, all right, the DNA is used to make a strand of mRNA. That mRNA is then ultimately going to go out into the cytoplasm and be used by the rRNA and the the tRNA, okay, to make the protein eventually. So here, after being made in the nucleus, the mRNA is going to go out to the cytoplasm. So the mRNA is kind of like, it's the instructions. So it's taken the information from DNA, turned it into an mRNA strand, which is then going to go out and help make the protein. The ribosomal RNA, okay, is 2 subunits, and like I said, basically what it's doing is it's creating a platform for this all to happen. And so there can be a lot of different, the ribosomes can be pretty big and pretty complex depending on what protein they want to make. Transfer RNA is the molecule that's actually going to bring the amino acid which is one of the reasons why getting your protein and people talking about different branch chain amino acids and stuff like that is that the amino acids are the building blocks for the proteins. And so the transfer RNA or tRNA actually brings the amino acid, okay, into the ribosome to help create the longer chain. So the first step to protein synthesis is transcription. So this is where the DNA is actually going to be converted into mRNA. This is very similar to DNA replication. The DNA opens up at the, it's got the replication forks, although they're not technically called replication forks because it's not replication, it's transcription. But the DNA opens up, you see, kind of in that bubble. All right. So you end up having the blue strand, which is the DNA, as your template strand, which is then used to create this pink strand, basically using almost exactly the same machinery. Okay, it uses RNA polymerase, it uses different enzymes, but using the same basic idea. Okay, and then using those base pairing rules. All right, if the RNA polymerase sees a T, it puts an A, sees an A, it puts a U, a G, it puts a C, and so on and so forth. Okay, so that's step one to transcription. So the first step of protein synthesis is to take the DNA, which isn't capable, for the most part, of leaving the nucleus, and converting it into mRNA so that mRNA can actually leave the nucleus and then can go do its job. I don't want to get into step two just yet. That will be your next lecture. I just think you might need a little bit of time to sit and digest all of this first. Okay? I'll talk to you later. Have a great day. Bye.